Good morning, everyone. It's a thrill to be here. Certainly we'll get to Boolean logic inside living cells, but my real goal is to get you to think about biology and our relationship with biology. You see on the screen here an amazing movie from Jesse Roston, where he's representing what we see in nature, right? Biology is this system. It exists on our planet. It predates humanity as we understand it. It's capable of organizing matter and information to create these beautiful, improbable patterns that just continue to promulgate and exist over time. Now, we as people, as humans, have developed different types of relationships with biology, and I want to give you a real quick tour of how we've experienced our interactions with biology over time. And if we think about it from an engineering perspective, not only as a scientific perspective, are we building a new biology? And if so, how do we wish that to develop? So for example, here are 6 instances of plants found in nature. That's a carrot, that's a watermelon, that's an eggplant, that's a banana, that's broccoli, and that's corn. They look different because these are the versions or instances of these natural plants before they were domesticated and bred for agricultural purposes. So corn, as found originally, doesn't have a cob, has a few kernels, and so on and so forth.
We really enjoy the ability to take the gene pool and use it like paints. It's our art. This is my art. I made this beautiful dog that I enjoy. I made her. I chose her sire and her dam. I chose several generations to make this beautiful dog. I'm very proud of her.
By observing the properties of plants and animals, of organisms, and selecting for traits we like, and crossing and breeding and repeating and repeating and repeating, we've changed the biology that we find originally. We also changed the biology by changing the environment within which the biology exists. Right? If we cut down a jungle and replace it with an oil palm plantation to get palm oil, then all of a sudden the animals that were living in the jungle are no longer living in a jungle. They might go away. Just to be clear, I'm not trying to take position on the right or wrong of this. I just want to give you a quick on-ramp to what's happening. Now, about 40 years ago, scientists discovered mechanisms, molecular mechanisms, by which we could cut and paste genetic material, DNA, to make chimeric molecules. This is a patent issued in the United States from 1980, very famous patent, that is the first patent claiming the capacity to make what's called recombinant DNA. You take 2 pieces of DNA found in nature, you cut them out, you paste them back together, you have a new chimeric molecule of DNA. This technology went to market starting at around 1980, and it's deployed in our economy. So in the United States today, the estimates from Rob Carlson, as published in Nature Biotechnology, suggest that the genetically engineered US domestic product, if you will, is somewhere north of $300 going on $400 billion a year in revenue. The major areas of products are biologics, which means medicines, industrial enzymes, and not without controversy, GM crops. And you can see the growth profile over time. It's one of the fastest growing sectors of our domestic economy. To give you a sense of scale, this is more than mining, extracting stuff from the crust of the earth. So within a few decades, a relatively new tool built on top of natural biology has gone to market, is now a significant component of our society. What does it look like to make stuff with biology? In the purple, bluish gray, you can see some microbial cells. They've been engineered to make an oil-like substance. You feed them sugar. The sugar might come from sugarcane, and out comes an oil, which could go into a fuel or a skin cream, or so on and so forth. Here's my wife. Last year, she and her team took baker's yeast and added 30 enzymes to that, plus or minus, such that those yeast could make opiates. You feed sugar to a fermenter, and out comes hydrocodone, a painkiller, or something else you might otherwise source from a field of opium poppies. Not without controversy to think about changing the material supply chain for a class of natural products that's otherwise quite secured and abused and has an illicit market. So why should that turn into a company and go to market right away? Well, it turns out that over 5 billion people on Earth don't have access to painkillers, and the World Health Organization considers these to be essential medicines. In the plant systems, it's very hard to change the molecules that are being made, because it's not easy to engineer the plant. But in yeast, if you have control of all the biochemistry, maybe you can make alternate molecules which have the pain-killing features but less addictive properties. Unclear if that's possible, but at least now we have a chance to go after that. So we'll see if they can do that. Dennis in Hawaii, papaya falling victim in cultivation to a virus, the ring spot virus. In a type of homegrown genetic engineering project long ago, he basically created a genetically engineered vaccine for the papaya plant so that it could now survive infection with the ring spot virus. So I just want to give you a sense of things that are happening, because what's coming next is a question and an early answer to the question. So the question is, well, we've been going at this for a couple decades. How good could we get at engineering biology? Or as we're talking about it, how good could we get at engineering living matter? And what I'm going to try and represent to you is, we think the answer to this is approximately incredibly good. Like as good at engineering living matter as you might think about our capacity to engineer a bridge or a computer or other things that we're quite familiar with. So what's shown here, for example, is a strategic document. It's a public document from the engineering school at Stanford. It's not my document. It's the whole engineering school at Stanford saying, this is one of the most important things we could be doing over the next few decades, because we think the answer is we can get really good at it, and we think it matters. Okay. Why do we think we can get really good at it? Well, here's an example of what it was like to get really good at engineering electronics. Lynn Conway and colleagues back at MIT in the 1970s said, how could we get a lot better at engineering integrated circuits? We haven't got the microprocessor yet. We don't have VLSI, very large scale integrated electronics. So they ran some courses with students and basically figured out how to do that. In 2003, as bioengineers, as would-be bioengineers, we tracked down Lynn and other colleagues who are expert in engineering transitions, and we ran a study for 18 months that was funded by DARPA, the US government's agency who's in the business of preventing technical surprise. And we were tasked with, could you get biology to be more engineerable, and what would you recommend we do? And here's the report. You can study the whole document free online from that link. Let me just give you the cartoon summary of what we recommended. Any type of engineer, once you figure out and choose what you want to build, you're going to have to design the first prototype, then you're going to have to make the prototype, then you're going to have to test your prototype and did it work, and when it doesn't work, debug it and rebuild it, and keep going around this cycle of design, build, test. So if you want to get better, if you want to get systematically better at the engineering of some new type of material, what we thought you should do is look at the core of the engineering process, design, build, test, and figure out how to upgrade navigating that process. Can you go around this process more quickly? Can you get more people to go around this process? Can you go around this process more smartly so that your first designs always work? Can you build more complicated systems by managing complexity? And so in the context of living matter, we said this new tool, which I'll get to in a minute, called DNA synthesis is going to be critical because it's going to allow us to separate design and construction. Some architect designed this building. Some other contractors made the building. They were each experts in their profession. We also said that you should develop technical standards that support coordination of labor so that what you might make in your kitchen or garage or laboratory, you could use later on in a different project. So you get reuse and building upon accomplishments. And then lastly, this abstract concept called abstraction. Everybody knows biology is really complicated, and so we can't remember everything in every moment. So we have to have some way of managing the complexity of biology. This is what we said back in 2003. And since then, we've been working on it. And the results from working on it suggest that these turn out to be really good ideas, and we can keep going on them. So let me give you a tour of what this means. What a nice photo. Here's a question for you. If you wanted to build a computer from the raw natural materials you see in this photo, what would you pick? Sand. Why is that? Silicon. Very good. Now, would you expect you could take my 3-year-old boy with you to the beach and a little pail and a scoop and get a scoop of sand and bring it over here and we'd make a computer from that material? No chance. Because the sand as we find it in nature has not been regularized, has not been made easy to work with, has not been organized in ways that's a composition of new improbable patterns and objects like a microprocessor. It's very normal for some reason now to think that, well, yeah, I take the sand and then I'm going to have to do a whole bunch of stuff to it, and eventually I'll get something that goes into a computer. Keep that way of thinking in mind when you approach biology with the mindset of an engineer. Why would I try and build stuff with the things I find? I'm going to have to figure out how to refine them and change the materials and make something more easier to work with. Okay, well, so I'm not an electrical engineer today, I'm a bioengineer, and so I can't talk about building a computer from the sand. That'd be a different lecture. I want to talk about building a computer from the biology in this photograph. And the biology you see is the palm tree, but I'm going to talk about the biology you can't see, which is the bacterial viruses that infect the microbes in the ocean and in our guts and everywhere. So here's a movie we made back in 2008. This is a time-lapse microscopy movie of bacterial cells, E. coli, about a micron long. Those are those oblong black blobs. The cells that turn green have been infected with a virus. So you can see that 2 cells have been infected. A cell in the top right of the movie, it just turns green and then it goes away. And it goes away because there's a light microscope, and when it bursts and the virus releases out, you can no longer see with this microscope what's happening. But if you take a different microscope that has better resolution, you can see the virus particles bursting out of the bacteria. But go back to the movie on the left. The cell in the top right, it turns green and then it pops or lyses. The virus is released. But check out that other cell that turns green down in the bottom middle left. It's green. Still green, divides, and keeps growing. All the other cells that don't turn green, they've not been infected. So it's only those 2 cells. Now what's interesting is that one cell that's infected pops and releases more virus into the environment. The other cell that's infected doesn't, it goes dormant. One possibility or another possibility. 1 or 0. There's a digital outcome here. All or nothing. Well, that's kind of interesting. It implies that there's a switch here, which is well understood in biology. This is bacteriophage lambda and its classic cell-fate selection process. But to an engineer, this implies I can implement digital decision-making inside synthetic genetic systems. As a subtle point in this movie, before we did this work, people thought that when virus infected cells, you couldn't tell ahead of time which cell was going to pop and which cell was going to go dormant. That it was just random. That it's sort of like the virus would infect the cell and there's like a rolling of the dice or a flipping of the coin, that it was stochastic chemical kinetics. Francois St. Pierre, who did this work, a biology student at MIT, found that depending on how big the cell is at the time of infection, it's much more likely to pop or stay whole. So you might notice that the cell in the top right, when it turns green, is much bigger than the cell in the bottom left, which means it's more likely to lyse. And there's about a 16-fold biasing. So that meant not only could we expect to realize digital switching, but that it could be deterministic, not necessarily stochastic. This is a very inspiring result for us back in 2008. So we set out to make something that looks like this. This is the abstract representation of it. I'm not giving you all the complexity. I have a piece of double-stranded DNA on the chromosome, the genome of an organism somewhere, and I engineered this little element called the flippy. And I'm going to use the same enzymes found in these bacterial viruses, the phage, not to run a phage infection, but to do something different. I'm going to use them to flip the DNA around. Now, the natural process here is, remember those cells that go dormant? What that means is the virus goes into the cell and then integrates its DNA into the chromosome of the host, and then it just hides out. So it's got this molecular tool to take its genome, the virus genome, and put it into the bacterial chromosome. Long ago, biochemists figured out that you could change how those tools work, those natural tools, to do something different, to flip DNA. A predefined synthetic sequence of DNA, nothing coming from the virus at this point except the tool for flipping or manipulating the DNA. And if I could flip it in one direction, maybe I could then aspire to flip it back. And if I could do this, now imagine I'm basically flipping something that's a go signal, so that if this go signal is pointing to the right, I could turn on a gene that's off to the right. If I flip it to the left, I could turn on a different gene. In this simple example, the genes could be different colors, make the cell green or red or so on. Note that's a 1 or a 0 in an abstract representation. We did that, and we turned out to be the first people to figure out how to flip and flip back and flip and flip back and flip and flip back as many times as we want an arbitrary sequence of DNA. However, that's less interesting to me as an engineer than how we did it and the process by which we realized that object, right? Because remember, I want to upgrade the design build test cycle, because if I can figure out how to upgrade that cycle, then I can get systematically better at engineering living matter, and I can realize geometric improvements in my capacity to engineer life. And it turns out that when we did this work, it was not easy. It took Jerome Bonnet and colleagues working in the lab 3 years to make the world's first system to flip and flip back, flip and flip back, flip and flip back. And all we had to do was express a couple proteins at different points in time to different levels, and it still took us 3 years. And it took us 3 years because it took us 700 different design attempts. We had to try 700 different specific engineered DNA sequences to get the levels and the timing of the expression of the enzymes just right, so that it would flip when we wanted it to flip, but not flip when we didn't want it to flip, so it would flip back when we wanted to, okay? And this isn't an engineered artifact, this is a work of art. Here's the sequence of the DNA. Can you figure out where in the sequence, is it apparent what makes it a good biobit? I can't, and we did it. Right, so this isn't going to work, right? This isn't going to scale. I mean, we can get it to work once, but I'm not going to build something super sophisticated. Let's say I wanted to, look at the top of this diagram. I wanted to make an 8-bit counter. So I have 8 DNA elements that are flipping in combination. An 8-bit counter would let me count up to 2 to the 8th, or 256. Imagine in every cell of my body, I could have an 8-bit counter running, and every time a cell divides, I could increment the counter by 1. All of a sudden, I'd be tracking cell division and aging. I could reprogram development. I could study aging. As my cells get older, I could stop them from dividing further if they're becoming a tumor. Wow, like just 1 byte of data inside every cell of my body would be amazingly valuable. I don't want this to replace your cell phones. I want this to implement computing memory under my control inside systems where microprocessors will never work. How do I ever make something that, see, an electrical engineer would say an 8-bit counter is simple, but a bioengineer would say an 8-bit counter is incredibly impossibly complicated. How do I ever do that? What's shown on this figure is this notion of abstraction, which is adapted from computer programming. I'd like to be able to have a really complicated system that I can think about and design at a high level, and then compile it down to simpler objects that are easier to get off the shelf that work reliably as expected. So the next level down from the top would be this flippy. I need a whole bunch of those on the shelf somewhere. And how do I make flippies? I need a whole bunch of different enzymes for flipping DNA, and somebody better be expert in how those proteins bind DNA and do the flipping. And then somebody else better be able to get me the DNA. Right? I want to be the system engineer for life, and I don't need to know what DNA is made up of. Why would I need to know that DNA is made up of 4 bases? Maybe I've heard about that. But if I were to send my mom a text message from, hey, I had a great time at World Science today, here's a photograph, I'm not thinking about zeros and ones when I send that text message. Maybe I've heard that computers run on zeros and ones. I just want to send her the message with the photo. All right, so that's what abstraction's about. This is not something we discover in nature. This is something that we are attempting and realizing via engineering. It's very controversial. It's very difficult to do. You have to figure out the interfaces to get the coordination of labor right. And we've just begun to show that this is not impossible to make true. If you've heard about CRISPR as an example, the reason why that's so powerful is it has a built-in abstraction aspect to it. You just swap out the guide RNA and it works, right? But for most other types of biology, we have to implement via a process of refining the abstractions. Okay, so it turns out, though, once we learned how to do this, we could figure out reliably flipping whatever we wanted. I don't have to flip something that makes cells turn green and red. I could flip something different. So this funny little schematic shows this thing we call the transcriptor. The T is in the middle of a strand of DNA. This is called a transcription terminator. It's an object that when RNA polymerase, which reads out DNA, flows from left to right along the DNA, this transcription terminator will halt or stop the transcription or the flow of RNA polymerase, the flux of gene expression along the DNA. Now, if we flip that terminator, it's like a check valve. The polymerase flux, the transcription, the reading out of the DNA will continue, right? And I could flip it back and halt it again. So if I had genes before and after this object, The genes before would always be expressed. The genes after would be conditionally expressed depending on the state of the terminator, the T. That's it. We could make this really quickly because although it had taken us 3 years to get good at flipping DNA, we designed that in such a way that we could flip anything else. So we just swap out what it is we're flipping, and it works the first time. And we made a whole bunch of different sorts of things to flip. On the top left, you can see a schematic where we have 2 T's in series. This is going to be George Boole's logical AND function. For this to work, for the polymerase to pass through that top left object, I have to flip the first T, and I have to flip the second T. If I flip one and the other, I get pass-through. If it's cold and it's raining, I'll bundle up. If it's raining or it's snowing, I'll bundle up. If it's warm and it's sunny, I'll go to the beach. These funny little words, and or, and so on, are what Mr. Boole back in the 1850s in Ireland recognized through an analysis of language, basically, are just useful. We find these words, these operators, to be incredibly useful in interacting with what we observe and so on and so forth. And humans have implemented the equivalent of and, or, all these logical functions in every type of material we ever have engineered, in hydraulic systems, in mechanical systems, in electrical systems of our time. But now we're of the time where living matter becomes engineerable, and so we're implementing Mr. Boole's logic inside cells. This is what those things should behave when you have the 2 inputs. Now you might be asking, what are the 2 inputs here that are controlling the logic? It's whatever signal you can get into a cell. It could be a microbe in your gut. It's like, oh, do I have a lot of fructose in my gut? And do I have an immune system marker? If so, change the color of the bacteria coming out of me, as you'll see later. Right? So just, but whatever signals you want. Do I have arsenic in my tap water or lead in my tap water? In which case, change colors. Right? So logic in new places. We made these. They work. They all work the first time. Every DNA design shown here worked the first time. It took us 6 weeks to do this project, not 3 years. It took us longer to get the scientific paper published than it did to get the work done. And just to give you a sense of what this looks like, cells growing in the bottom, the control signal in the middle, and then the output signal of the gate on the top. An interesting technical aspect of these gates is that they're amplifiers. So the signal needed to flip the gate is tinier than the signal produced by the gate when it flips, which is super important if you need to relay signals into more complicated computing systems. Okay. So that was a hint, a taste of standards and abstraction. Let me spend a bit of time on DNA synthesis, because this is a big deal. And I'll admit, I'm hopelessly biased. In addition to being an academic, I've also started 2 companies over the last decade to get better at building DNA. So just want to let you know about that. What's shown here are 4 bottles. Since we're talking about biology, you could guess that the bottles contain the bases of DNA, A, T, C, G. That's what we've got. Bottles cost about $250 each. You might be asking, well, what DNA do they contain? Do they contain strawberry DNA or lizard DNA? No. Just raw DNA, not taken directly from any existing organism, synthetically produced starting from sugar and sugarcane and derivatized, and then the bases are made from scratch, and then they're produced in these bottles. What I'm going to do is hook these bottles up to a machine called a DNA synthesizer. DNA synthesizer is just like a musical synthesizer, a keyboard. All right, so think about synthesizing music. I have 88 keys on my keyboard. I play the keys. The music comes out. I hear the music. DNA synthesizer, just like that, except it only has 4 keys. A, T, C, G. Right? At least the simplest ones do. And what comes out is a physical molecule of DNA made from scratch. The molecule of DNA that comes out of this machine is the molecule specified by the information you put into the machine, how you play the 4-key keyboard. TA, TA, CGA, CTC, ACT, ATA, GGG, AGA. Right? That's what I want. You can memorize DNA sequences, by the way, by having them be your login passwords to computer systems. So that's a nice sort of synergy between computing on the web and biology. Now, what does this sequence do? You don't want to ask that question. You want to return to abstraction and say, well, I'm going to rely on some framework to keep track of what all the low-level sequences do. Again, I've said I'm biased. The reason I'm biased is because this technology allows me to go from abstract information on a computer somewhere to physical genetic material that can instantiate behaviors inside living systems. So I'm just going to declare that's really important, and I'll put that up as a candidate for the most important technology of our time, now and going forward. When I first started teaching, The cost of doing this for gene-length pieces of DNA, 1,000 or 4,000 bases long, was $4 a base. Last year, it was 4 cents a base. We dropped it 100-fold. By the way, if you're starting a business and your product price drops by 100-fold, that means you have to get 100 times more business to make the same amount of money. It's not a great way to make a lot of money. But it did make it possible for more people to afford the making of DNA from scratch. Where did we head? Well, the price of this in 2016 is already approaching a penny. So it seems like we have line of sight to cost down over the next 5 to 10 years, which is pretty good. So I've talked a little bit about these technologies. Let me sort of map them back into pacing, and this is where it gets a little bit strange. How do you figure out where things are going when some of the fundamental tools start to change? And all of a sudden, how you navigate that design, build, test cycle is really different. So this is the first class we taught in 2003. We had 16 students, $4 a base pair to build the DNA. No abstraction framework except as a very naive concept. Almost no standard biological parts except for what Tom Knights, who invented the original BioBrick standard, had chefed up, like 6 or 12 parts. Man, we could hardly get anything to work. Well, here's that same thing last year. It's now called iGEM, the International Genetically Engineered Machines Competition. It's like a genetic engineering Olympics. It's held every year. It's about 4,000 students in 300 countries. About 30,000 students have been through this. This is in the Heinz Convention Center in Boston. The way this works is at the beginning of the summer, high school students and college students get a kit of standard biological parts that have been contributed to from students in past year's competitions. So it's a give and get open source philosophy. And then wherever they are, they work on whatever projects they want to do. And so long as they're safe, and then they bring those projects and their results to this competition, the Jamboree, to celebrate and get stickers and prizes. It's totally awesome, and you should go or be part of it. It's actually amazing. I want to, though, get you to think about navigating these transitions, these exponentials, and how tricky it is. Right, so here's a very famous company, Pixar, that does computer-aided movies. We know of them today, but there's this amazing story of how many times they failed before they succeeded. So this great interview from one of the founders talking about this process. And this vignette has to do with the second time they tried to make a computer-animated movie in the mid-'80s. They had John Lasseter already ready to go, and the hero monkey and a dragon and whatever. And then he's talking about how, in this bottom paragraph, I discovered to my dismay that the computers were still too slow. The projected cost of animating the movie is too high and take too long, so we've got to kill the deal. It's the second time they had this experience. But because it was the second time they had this experience, they now had 2 data points for how much it would cost them to animate a feature-length movie, and so they could make a projection into the future when would be the right time to make the movie. And he says it would be 5 years from then, which is when they did Toy Story. Right? So isn't that interesting? What they were discovering as movie makers is known as Moore's Law. Features per chip, computing power, over time has been improving along an exponential where it's doubling. It's a log Y scale there, so this is transistor count per chip. Over my lifetime, right? And it's like, okay, well, we tried to make the movie in the mid-'80s. Nope, let's wait a little bit longer. Yep. So when Gretzky explains why he was a good hockey player, he says things like, I skate to where the puck is going to be, right? That's pretty smart. Now pucks, though, even on ice, experience friction, and so they slow down a little bit. It's actually kind of easy compared to, imagine you had a puck and it had a rocket on it, and it was accelerating across the ice. Now how do you skate to where the puck is going to be? You can still do it, but it's a little bit trickier. You have to understand that trend. Rob Carlson, again, has been trying to make sense of this for reading and writing of DNA. Sequencing, the reading of DNA, writing, the synthesis of DNA. On the left plot, in the blue, you see features per chip on microprocessors. That's Moore's Law. And then in red and yellow, you see our capacity to read and write DNA. Now, that's on a different axis. It's not transistors per chip. It's base pairs per day. How much DNA could you read or write? And then on the right plot is the cost down, the drop in pricing of doing this. So if you put this in the context of genomics, of sequencing, the first microbial genome, a few million base pairs, was sequenced in 1995, but then the draft of the human genome was sequenced in 2001, billions of base pairs. Did the geneticists get a billion times smarter during the Clinton administration? Unfortunately not, but the tool for sequencing DNA got a lot better, right? And so you could skate to where the puck was going to be as the puck has a rocket behind it. Huh. Well, that's interesting. Where's the puck going to be? Well, here was some fun project that showed up out of the blue. You could write a book that's a book, a piece of paper, and then you could turn it into a digital bit stream, and then you could turn that into a synthetic base stream. Right? So bits are just zeros and ones, but there's 4 bases, so in a sequence of DNA, each base could be 2 bits of data. Not biological data, just arbitrary abiotic data, like George's book. Right? I could store a whole library in a tube of synthetic DNA by taking the books in the library, making them digital information in zeros and ones, running that through some coding scheme into a DNA synthesizer and getting physical DNA, not as a biological molecule, but just as an abiotic tape for storage of arbitrary information. And that became possible for the first time a few years ago. George was working with my first PhD student, then a postdoc in his lab. Shri, shown in the bottom right of that last slide, now a professor of biochemistry at UCLA, is working with OKGo to put their albums into DNA. Because you can turn the music into bits and the bits into bases, and now you release your album in a little Eppendorf tube. Huh. Well, this is all a little bit strange. DuPont, for example, is a corporation in the United States. They make things that our civilization depends upon at scale. And as a company, they're over 200 years old, and they'd like to be 300 years old and still be a company. And so they're seeing what's going on in synthetic biology and wondering, well, what should we do? Right? They have 6 products on the market today that make money via biology. They make things via biology, and then those things go to market. Mostly they come from petrochemical-based refining and so on. They make a strategic decision that they have to move to flourish. When they're 300 years old, they have to move all of their manufacturing processes over to biology. How to do it? They come out and talk for 2 days, have an amazing conversation. And I'm not trying to explain this summary slide of the conversation other than to represent that it's complicated. And then one other thing. It's sort of like a snowflake on the tip of the iceberg. What that means is most of biology hasn't been discovered yet, and most of biotechnology hasn't been imagined yet, let alone made true. So how should you think about what we wish for and where we want to go? This is David Willits, recently retired as a cabinet member in the British government. He was in charge of research for all of England and also the university system. And making sure, for example, Britain is competitive as a nation in the future world economy and so on. Talked with him a couple times. An amazing conservative politician, has a nickname of 2 brains. He's like super smart. And his opinion was synthetic biology has huge potential. In fact, has said it will heal us and feed us and fuel us. That's the historical economy of food, medicines, and manufacturing. The UK can be world leading in this, and so that's why we're backing it with more money. All right. Well, what does that turn to when the government supports things? It turns into reports, and then it turns into projects. But let me give you a sense of the reports. So here in the United States, from the National Research Council, we have the Industrialization of Biology report. And it's a roadmap for how to advance the doing of the engineering of living matter. And then similarly in the United Kingdom, Richard Kitney and Paul Fremont, 2 leaders at Imperial College, write, what's going on, and this project is going to get us a new type of industrial revolution. All right. Is that what I want? Do I want to industrialize biology? When I scale up the making of things and stuff gets out of balance, and this is a photo of the sailing pavilion from the Beijing Olympics with agricultural runoff apparently leading to blossoming of stuff in the water. Provocative photo for sure. I'm not sure I want to industrialize biology, it turns out. I think I want to biologize industry, whatever that might mean. So if this was my original title, I'd like to walk you through a transition in my thinking with just a slight change, a few copy edits. what might this mean? What might it mean to figure out how to interface with nature a little bit differently? Does it make any sense? Or are we stuck with this 19th century of metaphor of industrialization when we look to living matter in the living world? Well, let's just look at a couple things. Since I've been born in 1970, the population of humans has doubled. Keeps going up for the time being. Apparently, since I've been born, the population of animals in the wild has dropped in half. Correlation? Okay. My colleagues in ecology and biodiversity publish articles that say things like this: accelerated modern human-induced species losses entering the 6th mass extinction. The previous 5 were not due to humans. This one's on us, they say. Since 1900, we've lost of order 70 mammal species, 400 other things, invertebrate family, so on and so forth. We can't see most of the other stuff yet, but we have no reason to expect it's doing better. Why is this happening? Changes in land use, invasive species, industrialization, toxins, some of the things we've touched upon. This is a really interesting photograph. I want to go visit this spot in India. It's a double-decker suspension bridge. It's grown in place from the roots of a rubber plant. It's passed down from generation to generation. It takes decades to form. But note that it fixes carbon as it grows. It gets stronger over time. It's associated with a culture of stewardship, right? And it might seem that a couple decades to make a bridge is a long time, but I can assure you in San Francisco, that's pretty normal now. But this is interesting for other reasons, because it reminds me of a couple things when you just start to do the back-of-the-envelope calculations like Elon Musk might as a physicist. Well, I've got this functional nanotechnology. It's not gray goo, it's green goo. It's taken over the Earth already. It's powered by photosynthesis primarily, and it's harvesting 90 terawatts of energy at any given moment on average through photosynthesis. Our civilization's running on less than 20 terawatts, so 90 is quite a bit of manufacturing power. It's got these properties of reproduction, of growth, of healing. It's massively functional if you think about how it connects with the periodic table, and it matters. It's got ramifications. All right. So what might I wish for? Should be able to provide for all of humanity. Should be able to stabilize and recover natural biodiversity. Humanity and nature should be able to flourish together. The back-of-the-envelope calculations suggest that there's enough on our planet to provide. We should take infectious and other diseases off the table. We're going to have to do that from a biosecurity strategy perspective regardless. We should do it from a public health perspective anyway. If we pull this off, let's go for not a culture of consumption. Let's go for a culture of citizenship. I don't want my kids to grow up being consumers of biotechnology. I want them to be citizens of biotechnology. And oh, by the way, as we do this, we'll learn a lot. We learn by building. So this has now, for me, become the aim. I don't want to get really good at engineering living matter for no particular end. I want to transition humanity from living on Earth to living with Earth. I don't know how to do it, because it's as much a cultural and economic transition as it is a technical one. The technical transition actually seems pretty straightforward. And when I don't know how to do something, what I do is I basically pay people who are professionals in the thing I don't know about to teach me something. So here's an example of this. We got $250,000 from the National Science Foundation, and we used that to create residencies, paired residencies between artists and designers and synthetic biologists. And the way it went was anybody in the world could apply, and if you get accepted, the artists spend a month in the laboratory, and then the scientist from the lab goes back to the studio. And do whatever you want. So we got about 100 people applying, and we supported 6 partnerships, or 12 people total. Sissel Talas, a perfume artist, a smell artist from Berlin, worked with Christina Agapakis, then a microbiologist at Harvard. Here Sissel is in Cambridge, Massachusetts. And Sissel shows up, and she's super interested in the odors of cheese. And there's a great cheese shop, Formaggio's up in Cambridge that ages their own cheeses. And so they went there for a visit just to smell. But of course, they brought test tubes. And they pulled core samples of the stinkiest cheeses to bring those back to the lab, because cheeses are made with microbes, so a microbiology lab would be a good place to study this. And you could see what grows out. Now, it turns out that as they're having conversations, they realize that some of the cheeses they get are these small batch artisanal cheeses that are made by humans. And they're also talking about what's exciting in the world of the science of microbiology. And the thing that's exciting is the human-associated microbes, the so-called microbiome, the things that live on our skin and in our guts, right, which is just becoming apparent to us in great detail because of DNA sequencing improvements. And they go, I'm going to put one and one together, and they go, what's the relationship between the microbes that live on our skin and the microbes that make our cheese? Especially these small batch cheeses, these artisanal cheeses. Now, a normal scientific response to the question today would be, let's sequence everything and then use a computer to align the sequences. But a synthetic response to the question would be different. Let's make some cheese. There's microbes all over us. What type of cheeses would they make? So they went around the Harvard campus with sterile Q-tips, and they asked their colleagues and friends for samples of their skin-associated microbes. And then they went to the farm and they got raw goat milk. This is all within a few weeks, right? It's amazing what professionals can do. And they started making cheeses. What would the cheese made from the microbe found on a philosopher's toe be like? How about from Sissel's hand or Christina's nose or vice versa, or Daisy's armpit? Right? I happened to be passing through Boston when they had done this. They showed up with a tray of 48 different cheeses, and nobody thought about or worked through biosafety, so we didn't eat them. But we just took a whiff. And I can report faithfully that Daisy's armpit cheese is beautiful. It's got a robust citrus floral bouquet, and I'd like to try it. Philosopher toe cheese is wrong. You would need to invent a new adjective to come up with a description of the bouquet of that object, and so on. What did this teach me? Well, growing up, my mom said, eat the green leafy vegetables, treat your body as a temple, you are what you eat. But this got me to reverse that. It says, no, we eat what we are. I've been disconnected from nature in my food supply for good reasons around public's health and food safety. But maybe there's a big cost associated with that. I need to learn how to reconnect. I was having dinner that night with a banker from Fidelity, and I thought I would open the conversation by trying to ruin his appetite by telling him about what I had just experienced. It had just the opposite response. This is a good example of why I'm so naive when it comes to figuring out how to realize cultural and economic transitions. What do you think the banker said? Billion-dollar market? Celebrity cheese? You could have political fundraising cheeses. Barack Obama blue, right? A washed rind red, and so on. Okay. Remember iGEM? Let me loop back to that and try and connect a couple things together. So in one of the earlier iGEM competitions, some folks come into the microbiology lab. Now they get to work on what they want, right? And they come into the microbiology lab, and they decide that it stinks. They don't like how it smells. It smells like E. coli, which is mostly the smell of indole, typically. So they decide to start a project called Eau de coli. Get rid of the indole biosynthesis, add in bioproduction for methyl salicylate, which gives you a wintergreen bouquet, add in production for isoamyl acetate, which gets you banana odorant, and then implement logic controlling the production of these two odorants so that when the cells are growing, they smell minty fresh, and when the cells have grown up and the culture is ready for you to harvest it, they smell like bananas. All right, so that's what they did. My mom asked, don't E. coli smell bad for a reason? Is this a good thing to be doing? She's probably right. What's that have to do with this? Think about what's implied by the object on the bottom right. I have centralized industrialized manufacturing, harvesting synthetic petro inputs, plant inputs, other things, into some factory. The stuff is chefed up. It's then put into bottles. The bottles are distributed and marketed globally through duty-free catalogs and everything else. And then people get this stuff and they open it up and we slather it on our skin. And for a few minutes, the volatile compounds come off and we like how we smell. That's what is implied by this advertisement. What if I could go to a website called Fumes, F-U-M-E-S, and for 99 cents, download the DNA sequences encoding the biosynthetic pathways of different odorants. And I had my local DNA printer, and I would print the DNA that makes those pathways, and I'd get that synthetic DNA, and I'd slather it on my skin. I'd transform my epidermal ecosystem so that I'd have living programmable perfume. I'd never have centralized manufacturing and then transportation costs. I'd ship the instructions as code over the internet and do localized distributed manufacturing. Of course we'd have to work out safety. Of course we'd have to work out acceptance. Of course we'd need to give Brad more money to market it, right? And do the advertisements. But just think a little bit about some of the unique properties of biology. It's a distributed manufacturing platform. It doesn't have to be centralized. Anywhere there's biology, you have a state-of-the-art nanotechnology that's programmable through DNA. The capacity to print DNA is improving exponentially. And so maybe we could change our material supply chains dramatically. Let me give you a different example. Another example from iGEM, this time from Cambridge, England. They were okay with the smell of E. coli. They didn't like how it looked. It's got this dull brown color, typically. So they did a project called Escherichia chromai. I wish they had called it Crayola. But basically, you take E. coli, you get it to make pigments, so that all of a sudden, under different conditions, the cell could be red or orange or dark yellow or light yellow or green or blue or purple, right? Living programmable pigments. What would you do with this? They didn't know. But they knew enough, just like I had learned, to go ask somebody who could think creatively. So they went to the Royal College of Art in London and asked the artists and designers, if we gave you living programmable pigments, what might you think people would wish for, whether they know it or not ahead of time? And the concept that came out from that engagement is shown here. You have a probiotic yogurt. The bacteria in the yogurt are instrumented with genetic sensors to monitor the disease state of your alimentary canal. If you're doing fine, nothing happens. But if you're a little bit out of whack, if you're missing vitamin A, if you've got an immune system marker, an irritable bowel syndrome is about to show up, activate the production of a different pigment so that what comes out of you is colored, indicating that you've got something going out of balance and going to maybe want to make an appointment to see the doctor. They called this the scatalogue. Now, this hasn't been made real, right? It's only been made real as an idea. And you'll see on the bottom left, they have a number in front of their product name, 2049. What's that? Is that the price they're estimating the scatalogue will cost? $20.49? No, that's the year in the future they believe we humans, our culture, our society, will be ready to accept such a product on the market. What's interesting is the yogurt companies, when they saw this, presumed it had all been made to work and were ready to bring it to market now. And it's kind of interesting because you could have a medical diagnostic platform that would be approximately free. It would be the cost of cultivating the yogurt such that anywhere you have raw milk and could get the microbes, you could have people maybe getting better information about the state of their health. And you could take this a step further and have the microbes not just report on what's going on, but maybe try and take some prescriptive action. So just a couple more examples. I have a tree like this in my front yard in Menlo Park. It grows pine cones. I didn't pay too much attention to it until I started thinking about the interface I have with life in nature. What happens is these pine cones grow every year, and then when the winds come towards the winter, they fall off, and then the things get raked away, and we put it somewhere, and it gets composted. The whole city is doing this year after year after year. And it turns out we're doing it to the tune of 500 pounds per person per year of garden clippings. In a city of 32,000 people, that's 16 million pounds of matter compiled from the atmosphere every year that we pay to get rid of and just compost. I have a self-assembling state-of-the-art nanotechnology in my front yard that I'm paying somebody to get rid of. This is crazy. What could I do with 16 million pounds of matter a year? Well, if I'm making a bulk commodity product like diesel fuel, not going to have a big impact. But what if I wanted to make a computer chip? Not a genetic computer chip, but a silicon computer chip. Well, there's about 1 billion chips sold a year. Let's say they weigh a gram each. That's about 2 million pounds of matter a year. The manufacturing capacity of the yards and gardens of Menlo Park, in the middle of Silicon Valley, is 8 times greater than needed to make the world's chip supply. how do I reprogram my pine tree to make a cash crop? How do I reprogram my pine tree to make microprocessors? I have no idea, and that probably doesn't make any sense anytime soon. I need to think a little bit differently about what I might do with this material. And the lesson for how to think differently about this came from an artist who I'll introduce you to, and also an entrepreneur, a fellow named Phil Ross. Phil introduced me in a new way to these things we're familiar with called mushrooms, the wood fungus. We went to go visit a farm called Far West Fungi, which has their operation inset within the strawberry fields near Monterey, California. Here's Phil and I visiting the farm. The input to the farm is piles of sawdust, just agricultural waste approximately. There's a pile of sawdust. They then take the sawdust, add a little bit of moisture to it, some crushed oyster shells, put it in bags, and then sterilize it in a big pressure cooker. There's the son of the farmer. The bags come out on a belt. You have a little inoculum, a liquid inoculum of a mushroom culture. The mushroom starts to convert the cellulosic material into mycelium. It then gets racked, and these loafs of mycelium then have fruiting bodies of the mushrooms that come out. Depending on the strain, you get different things fruiting out. They're harvested, sorted, and brought to market as California-grown organic mushrooms. This is one of the best suppliers of mushrooms in the farmers markets on the San Francisco Peninsula. We're eating sawdust. It's wonderful. Phil recognized, and others, that you can take these organisms and use them not just to make mushrooms that we might eat, but the fundamental conversion from the cellulose to the chitin in the mycelial matrix just lets you make stuff. And the stuff that gets made is formed by a form or the explicit shaping of how the sawdust is packed. So I could grow a donut or a wheel. I could grow big biobricks and stack them together to make a tea house. And I could actually make tea from the tea house by breaking off a little piece of one of this and dissolving it in hot water and drinking that. Or I could make a chair. Or I could make children's toys. Now, what's interesting about these children's toys, if you let them be dormant, you don't fully sterilize the blocks, and then you reactivate it with a little bit of moisture, once it's assembled, you could then have it grow and fuse into a continuous object. Huh. It turns out with these organisms, you can make many, many different types of materials. The wood fungus are amazing, and I would expect to see many examples coming out this year of things you'd never imagine a mushroom could make showing up in places you wouldn't expect to find it, on the fashion runways, for example. So let me end there and return to this slide. On the left, we've got this amazing thing on our planet. It's called biology. It actually has the capacity, in my opinion, based on back-of-the-envelope calculations, to provide enough for nature and for humanity. But we're not well configured to interface with it. We have to figure out how to take these two things, and maybe we need to change what we wish for. I'm only representing my struggle to articulate a telos, or primary aim, on the right. So there's going to be a diversity of aims, to be sure. But whatever that turns out to be, so we have to figure out how to bring those together. Right? Biology exists. We're now on a journey of getting systematically better at engineering living matter. The evidence supports over the last decade and a half that we're going to pull it off. We're going to pull it off within the next 20 to 50 years, like going from computing in 1950 to computing today, probably even a little bit faster. What do we want? Thanks very much.